Thank you for joining us today, everyone, and welcome to this drug discovery at superluminal speeds. Today's session will examine how integrated computational platforms accelerate workflows from virtual screening through lead optimization, with an emphasis on Superluminal's Hyperloop platform and its interoperability with Schrödinger's LiveDesign and CDD Vault. We will focus on architecture and experimental validation, concrete use cases to show measurable gains in discovery timelines. So we have an excellent speaker panel planned for y'all today. Our first speaker is Ajay Yakirada, co-founder and SVP, head of discovery and development at Superluminal Medicines. We also have Mark Fitzgerald, also from Superluminal Medicines, director of chemistry. Next, we have Kevin Halligan, vice president at Schrödinger, and James White, director of collaborative communications at CDD Vault. We'll begin with brief presentations followed by Q&A. Please submit questions at any time via the Q&A toolbar in your Zoom. With that, Ajay, thank you for joining us. The floor is yours.
Thank you so much for the introduction and for getting us going on this. Folks, we're really excited to actually give you a sense of what we're doing at Superluminal and how integral CDD and Schrödinger have been to kind of the success at Superluminal as well. So to just give you a sense of what we are up to at Superluminal and what Superluminal really stands for, we are a very structure and biology and chemistry focused company that uses computation as a way to leverage and accelerate the things that we do. But at the outset, we're very experimentally focused. And I think I want to belabor that because this is the era of a lot of hype around AI and machine learning based platforms. We use a lot of AI and machine learning based approaches. But the whole aspect is to really focus on biology, chemistry problems, and seeing where potentially machine learning and computation at scale could really provide solutions. So that's at the core of the ethos of Superluminal. From a target class perspective, we have been focusing on GPCRs. And I'll explain why we kind of went to this particular target class right at the beginning. But the key point is we are very heavily protein dynamics focused, and I'll kind of touch on that as we talk about our platform. Fundamentally, at Superluminal, we've also been able to bring on a terrific group of investors through our seed and Series A raises, and we've raised about 153 million now in total capital from RA Capital, Insight Partners, NVIDIA, Catalio, Eli Lilly, Gangels, and Cooley. And we're really fortunate to have all these folks kind of be with us on this wonderful journey. So as I mentioned, we're extremely structure and biology focused, with chemistry being the real tooling there. And the idea is to use the insights that we gain from structures and move very, very quickly to experimental validation. The whole aspect is to really break this down into experiments so that we can validate all our hypotheses as quickly as possible. Computation is there to allow for acceleration and to also allow us to do some very unique things where maybe the biology or the chemistry state of the art is not there yet. And I'll touch on like fundamental aspects of how those pieces are built together. But the key point is this: the computation is there to serve the needs and problems and questions in biology and chemistry. And that's really the focus at Superluminal. At Superluminal, we're really focusing on three fundamental areas to really bring that computation to bear. The first aspect is proteins are inherently dynamic. They cycle through several conformations, but whether it's through crystallography or cryo-EM strategies, we're typically only able to get images of the most stable conformations. And we also have this chicken and the egg issue where unless there is a ligand stabilizing a given conformation, we may never see that. Even if we get apo structures, we're fundamentally seeing the most stable conformations. And then the final piece is we make a lot of modifications to the native sequence. And for membrane proteins, for instance, adding things like Brill tags and fusion proteins, et cetera, to be able to even resolve the structure. So there is an open question as to what exactly we are seeing. Most importantly, are we developing drugs for the right therapeutic conformation of a given target? That's the real question that drove us. So that's where we've started utilizing machine learning based approaches where with generative AI, let's say with AlphaFold, we as a field have taught algorithms to fold a sequence into a given structure. But we are trying to take it to that next stage where can we teach the physics of movement to these algorithms as well? And if we think about this, we know the sequence already. It's all about bond angles and bond lengths. So the real idea is to really generate this kind of a coordinate density map, if you will, of all the possible states that that protein can exist in. And if we can generate that, then we can map trajectories of motion. And along those trajectories, we can sample conformations. So what this gives us is an ability to really get a high definition understanding of all possible states that protein can exist in. Now comes the next question. You've got all these conformations. Let's say I have 10 conformations for a given target. Which of those are real? And which of those are actually therapeutically relevant? That's where we felt there are two different ways we can get at it. One aspect is to throw the kitchen sink, identify conformation-specific ligands by really exposing these conformations to ultra-large libraries. The current theoretically accessible chemical space is thought to be something like 5 to 7 trillion molecules. These are enumerated libraries like ExploreDB for e-molecules, the Wuxi database, Enamine's RealDB, etc. You put it all together. No one synthesized every single one of these molecules, but these are enumerated. So you can theoretically throw these virtual compounds onto these conformations. But even screening 50 billion compounds is actually a pretty significant exercise in silico. Others in third-party groups have actually already tested this out, and with significant computation, just Enamine's 50 billion compounds would take around 4 years to get through using any docking software. So that's where we've again taught machine learning-based algorithms to really understand the physics of docking. And we do this in a very conformation-specific way. So each conformation aspect is embedded into this docking exercise. So you have each model is for one particular conformation. And so that can then get exposed to these large libraries. And we are now able to screen the same 50 billion compounds, for instance, in less than 10 hours. So what this gives you is the ability to really travel across these large libraries and to do it in a sizable time that really allows for discoveries to be made. And because we don't have license limitations, etc., you can actually now screen for all the conformations in parallel. That allows us to also have the right throughput there. A second way that we can get at this is to use three-dimensional pocket to molecule type approaches, which we also very readily use. Obviously, the chemistry that is involved post-fact to build such molecules tends to be more complicated because you are not dealing with easily enumerated library type molecules there. But we use those strategies as well where we need that. And the final then piece that comes to is you can generate all these molecules or you have screened these large libraries. Let's say we have 10,000 potential in silico hits per conformation. We don't want to synthesize 10 times 10,000 compounds. That would be a lot of effort. Instead, we've built these druggability filters where we have taught algorithms to understand ADME-tox liabilities. And we did this with both public data sets, which are not that great, but we've made the most of them, and also getting access to proprietary data sets. One of those was a pharma data set, 200,000 plus compounds across 31 ADME-tox parameters. All of your usual suspects from liver tox to gene tox to cardiotox, hERG, blood-brain barrier penetration, etc. What this gives are decent models to which you can throw all these in silico hits and get a dashboard of potential liabilities. Anything egregious you can triage. Anything that you can use, you can definitely move towards synthesis. So that fundamentally describes the platform in of itself. To get then a bit more into the structural biology piece, because remember I started off, we are very experimentally focused. In as much as we generate a lot of models and understand dynamics, we are also very, very, very keen that we have significant structure capabilities. So what we've done here is to take a very decentralized approach. We have access to 13 microscopes at 6 different sites across geographies. And the idea really here is to diversify both for logistics and also for capabilities. Not everywhere you're going to push for a 2.5 angstrom structure. Some places you can really screen at 3.5 angstrom, 4 angstroms, just very quickly, multiple sets of compounds to see what would be giving us a good structure, and then move it to other places where we can get higher resolution with water molecules, etc. So it's really about trying to maximize our potential there. We've also been pioneering machine learning-based approaches both for acquisition and then processing of the data. In fact, the acquisition piece has been going really well, where we actually use our GCP-based platform to hack into these microscopes. So all someone needs to do at any of these sites is to really load the grid, walk away. It's our scientists who are then acquiring the data, and the machine learning-based approaches allows us to acquire as much as 5 times the data as a manual setup would. What this allows is very quick, options for us to get to all atom models. So we've been generating something like, 10 plus crime structures every month. And we have the capacity actually to get to the throughput of 15 to 20 structures a week, just as much as we need it. In fact, for our lead program that we just declared our development candidate for MC4, we've generated, something like 27 to 28 proprietary structures, even with non-canonical G proteins, etc. So really generating a very clear structural understanding of this target. And just to give you a sense, in PDB, there are only 7 structures of MC4. That just goes to show how much structural work we do. Most of that is to validate our models and also to give high definition structures to do FEP and other strategies, QSAR strategies for lead op. All right, so the Hyperloop platform itself, just to delve a little bit more in depth there. As I mentioned, we use a combination of several enumerated libraries, which we can say, the space is very, very large. It's probably north of 7 trillion ligands now. And every day seems like, more ligands are added to these libraries. And we have predicted and also experimentally generated conformations. All of these then go together, and we use in the hit stage a combination of the machine learning based gigascale screening approaches along with the ADME-tox predictions to very quickly get to compounds that we can synthesize, which then go into screening assays. And if we are able to then understand the pharmacology and match up, let's say, a given conformation to a particular, pharmacological endpoint, then those then go into cranium to, also validate and get structures. Now, this is really the space where we started really looking at GPCRs as the key target class for superluminal. The fact that we can generate all these conformations is really valuable to go after this because it's no longer enough to generate just agonists or antagonists of GPCRs. Really talking about biased agonism, biased antagonism. What do I mean by that? G protein coupled receptors coupled to several different G proteins and also beta arrestins, amongst other things. Each of those leads to a fundamentally different pharmacological context. So physiology matters. And in as much as we know a particular pathway that we want to activate for a given target, for instance, for MC4R, we really wanted to build bias towards the GQ pathway for XYZ reasons that I don't want to get into right now, but pharmacologically, we knew that was important, but no one had solved a GQ conformation for MC4R. So we used these conformation-based approaches, generated several conformations, and we were able to actually synthesize compounds that were conformation specific. And in screening assays, which were functional, we could tell, oh, it's actually conformations 4 and 5 that are giving a lot more GQ biased, signals, whereas conformations 1 and 2, for instance, giving us more GS biased signals. So that's how we learned. The pharmacology and the experimental profiles actually tell us what those conformations mean, if that makes sense. And then we took those compounds and then we were able to generate using cryo-EM structures of MC4R, let's say coupled to GQ and so on. So that's kind of how that entire piece comes together. This happens very rapidly. And from there, we then transition more into the lead stage, where again, we use, as I mentioned, the de novo design type strategies where using generative AI, you're kind of teaching, these algorithms to act like medicinal chemists. I know Mark is here on the call. It's to really give someone like Mark the tools to be able to get multiple design options very, very quickly. In fact, some of our tools are able to give hundreds of thousands of design ideas in a matter of milliseconds. So it's not that all of those are synthesizable. Let's not hype these things up. But from a reality perspective, you would need an army of medicinal chemists to come up with even 1/10 of those numbers of designs. So it's really about accelerating that and giving really brilliant chemists like Mark the ability to then use those designs features into building in their own SAR. So that's really what we do. In vitro and in vivo, iPSC-derived secondary mechanism, strategies, all of those are embedded into our wet lab capabilities, and cryo-EM structure-based efforts and biochemical efforts are always at the core of all that. And the whole idea is then to keep proceeding this way and use all of these capabilities to get to a development candidate in really, really quick time. But the key point I want to mention is that speed isn't everything in discovery. You really want to also have quality. And what we're ensuring from day one is that we're really building highly bespoke molecules focused on a specific conformation. So they're already having certain optimizations built in because we're already starting with the right conformation. We have built-in druggability along the way, so we're also having much better molecules to work with. And what this helps with, along with cryo-EM-based approaches, where we are really learning about what amino acid residues we are hitting, etc., it gives us the full landscape to reduce those DMTA cycles and speed up time. So it's both quality and speed at the same time. So what have we been able to do? Just to give you a sense, we first took on 6 different GPCRs. In fact, 4 of those were Class B GPCRs, really known to be difficult to drug. But we were able to actually achieve hit to lead for all of those targets in less than 5 months each. So just to give you a sense of how quickly we've been able to move. We can stabilize and understand specific protein conformations for these highly dynamic membrane receptors. So we have ambitions to go to other membrane targets at some point as well because of these capabilities. And it's really about trying to get this chemical diversity in place so that we have significant IP to work with. And more importantly, you also have druggability really baked in. So you're really building the right type of molecules. So that's the focus and how we've been able to kind of progress with this. We just declared our first development candidate, as I mentioned as well, and we're moving along on the rest of the pipeline. With that, all of this, by the way, wouldn't be possible if we didn't have the right partners. And I think here we've been very, very successful because of the fantastic partnerships we have both with CDD and also with Schrödinger. And to really talk about these, let's bring on the experts. So I would now transition this to Kevin from Schrödinger, and he'll take over from here. Thank you.
Thank you, Ajay. I am Kevin Halligan, a vice president in Schrödinger's sales organization. For the next few minutes, I will be discussing the LiveDesign platform. First, I want to extend my gratitude to the Superluminal and CDD Vault teams for joining us in this joint webinar with Schrödinger. Many biotech companies, much like Superluminal, leverage both the LiveDesign and CDD Vault platforms. They do this to harness the clear synergies between the two in order to push their discovery programs forward. This slide you're looking at here is a high-level overview of the entire Schrödinger computational platform. The platform integrates both computational biophysics and AI by leveraging the inherent complementarity between the two approaches. Physics-based methods rooted in first principles don't require training data, can extrapolate into novel chemical space, and are highly accurate. However, they are slower and computationally intensive. Conversely, on the other side of the slide, there's AI ML methods, which excel at speed and efficiency when handling large data sets and are effective for interpolation. A significant challenge for AI ML methods in drug discovery is their need for massive training sets of accurate, structured data, which unfortunately is often unavailable. To overcome this, the Schrödinger platform combines both approaches. It uses highly accurate physics-based computational modeling to generate virtual project-specific data, and then uses this data to train the AI ML models. This hybrid approach enables fast, accurate, and large-scale exploration of chemical space. Using this type of modern computationally driven approach to drug discovery does create a problem. What do you do with all of this virtual data? How do you scale access to these predictions across project teams? And even how do you enable all project team members to easily run the predictive models themselves? LiveDesign, which is mentioned at the bottom of this slide, is the collaborative enterprise informatics component of the Schrödinger platform, and it addresses this challenge. It integrates digital and experimental data side by side, facilitating access to all that data, both real and virtual. LiveDesign democratizes the ability for all project team members to contribute design ideas and execute any type of predictive computational model, whether physics-based or AI-based. So, what is LiveDesign? LiveDesign facilitates a predict-first, data-driven approach to drug discovery. It empowers team members to leverage robust informatics and data analysis, execute calculations at scale, or on individual design ideas, using self-service predictive models. LiveDesign also enables monitoring the progress of these virtual ideas as they move through the pipeline into synthesized compounds, essentially navigating the entire DMTA cycle, or as Schrödinger prefers to call it, the DPMTA cycle: design, predict, make, test, analyze. By fostering real-time collaboration across entire teams, LiveDesign drives projects forward. And this is my final slide, and it's a rapid video showcasing LiveDesign in action. Rather than attempt to narrate this, I will just offer a general overview of what you're observing. The user here is actively designing a novel small molecule compound within the receptor's binding pocket. This design process can be carried out in either 2D or 3D, with real-time predicted properties guiding the design. Following this, the user accesses an alternative data view within the live report, known as a form view. This view features customizable tiles, which in this instance are used to assess how effectively virtual and real compounds cover chemical space and to analyze structure-activity relationships. It's important to note that all views within LiveDesign are fully interactive. So that's it. But before concluding, I want to emphasize that while I've discussed accessing and viewing experimental data within LiveDesign, this data must come from somewhere. And a really great platform for managing your experimental data is CDD Vault. With that, I will pass the baton to James White, who will elaborate on CDD Vault. Thank you very much.
Thank you, Kevin. I'm really excited to be here to talk to you all about CDD Vault. So if you join me at a 10,000-foot view, CDD Vault emerged out of the fact that modern workflows are becoming increasingly complex. So even if we take a deceptively simple example here where you want to synthesize some compounds, do purity, run some assays so that you can do some modeling, right? Each of these is going to have their own file extensions that's being generated. And when you have to repeat this cycle 1,000, 5,000 times to create enough data to then do your modeling or do your drug discovery, you create a huge amount of data that you need to keep track of. This forms data silos, and moving information through this workflow is clunky and slow. And these data silos scale with an organization. So if your individuals are having to pass data between each other, that's slow, introduces version control issues. This scales further if you're working with collaborators. So if you have multiple CROs, do you know where all your data is? Is it the correct version? How is it being tracked? So CDD Vault simplifies this complexity and uses chemistry as the foundation for your entire workflow. These scattered files will fall away, revealing a single source of truth where your data becomes the interoperable hub of your entire workflow, securely stored within your CDD Vault environment. So I'm going to talk about our core functionality in the blue at the bottom here and one of our add-on functions today. So with the registration module, what we're going to do is replace those disjointed records from our original workflow with a chemically aware registration system. You can draw molecule by molecule within CDD Vault or import SMILE strings or structured data files within registration. We're going to ask, is this a new molecule or have I seen it before? If it's a new molecule, we're going to create a new entity. And if we've seen it before, we're going to create a new batch rather than creating a duplicate. This is a framework upon which we can now build and pull in data. So for pulling in data, that's where assays come in. We're going to be able to define the required fields for each experiment or each assay that you're running. So this is built with protocol forms. So you can build out the required fields for each experiment using common assay templates, which are ontology based. So we're also collecting rich metadata. What these look like. So if you're doing cell viability assays, we can collect specific information for each of those, flow cytometry, or even ELISA. These are fully customizable to your needs. So please reach out to us and ask if we can support your specific assay, and we will be able to. These allow you to capture complete records every time an experiment is performed, so you can get meaningful results. Within the activity module, right, where we can start to work with the data that we're collecting. What this looks like is a framework upon which we can build to analyze the data. So we've uploaded some raw data here. We're calculating IC50s, the hill slope, and we can also plot concentration inhibition. But importantly, this is a framework so that when we add in batch 2, these properties are being automatically populated for us rather than us having to do them ourselves. At scale, this looks like having all of your molecules arranged on the left and all the data associated with that molecule moving across to the right. Because this can be a huge amount of information, we have the visualization module where we can create custom data comparisons. So if we want to look at molecular properties with in vitro data in this particular experiment, we can do that and then get automatic project insights. So we can segment the legend by substructure or molecular weights, and as new data is added into CDD Vault, these graphs will automatically update. They can also be shared with colleagues to allow for seamless collaboration, so you don't have to keep generating new PowerPoints over and over again. You can just double check and work with each other directly within these visualizations. So this allows, with activity and visualization, to glean automatic insights, speeding up your workflow. But lastly, I want to talk about what we want to do when we truly can bring together an automatic workflow with automation. So this is where we can use our RESTful API to connect to external tools. So the data from your equipment can be automatically parsed and mapped into CDD Vaults, where it then becomes available to get exported out and connected to your external tools, say if you're doing some modeling. So this allows CDD Vault to truly become the single source of truth for your entire workflow, going from the equipment that's generating the data, storing it securely within CDD Vault, and then sending it off to external tools, for instance, modeling experiments. So with that, I want to turn it over to Mark at Superluminal to talk about specifics of how they use CDD Vault.
Thanks, James. I wanted to now dig in a little deeper into some of the virtual screening and triage that we do at Superluminal. And namely to mention that, there's a lot of hands basically that the data goes through, and it's not always the same tool that people use. And for instance, when we go from the virtual screening in the hands of the machine learning scientists, and then it's handed off to the computational chemists for triage, they often take it down from, tens of thousands potentially of hits to maybe thousands or hundreds, utilizing a variety of tools. And then we found that LiveDesign is a really convenient tool for whittling down these thousands of hits to more like hundreds of diverse chemotypes with potentially favorable properties that we would pick, for synthesis and then experimental screening. And I think it's worth noting that we found with our multiple projects where we've used LiveDesign that this is convenient and useful, not only in this, hit finding approach, but also later on in the kind of analog design and data capture. As I kind of denote on the left bottom segment, just to say that, hey, after we have the hits and we test them, the virtual hits, and we test them and have biological data that repeats, then we really are off to the races utilizing LiveDesign for both data digest as well as design and tracking of activity. And just from the bullets here, I'd like to highlight, that really this is a tool that's not only useful for chemists and comp chemists, but we have the biologists using it at Superluminal as well. So it's really kind of a nice common place where everyone can, access the data and contribute to the project goals. And with that, I'd like to speak a little bit too of our use of CDD at Superluminal. And in addition to chemical registration, which we know and love CDD for, we also utilize their vault for the primary storage of biological assay data. And we basically are able to, utilizing the API that James spoke to, we are able to bring in a variety of data from a variety of sources. And then it's basically calculated and curves are generated automatically upon upload, which really helps with the cycle time and kind of getting the curves, really the kind of QC data to the hands of both the biologists and the chemists. And with that, I'd like to thank you for your time. And it was, my pleasure to introduce some of the uses of these tools at Superluminal. And with that, I'll hand it off to Josh.
Thank you, Mark, and thank you to all of our panelists for your fantastic presentations today. We'll move on to Q&A shortly, but before we do that, we'd like to run a quick poll. If you're interested in learning more about what you saw today or even scheduling a demo, please vote below what you would like. A representative from the respective organization will follow up with you directly after the webinar. We will also be sharing each organization's website as well as social media in the chat if you'd like to explore at your own pace. Now, while y'all are filling out the poll, let's move on to Q&A. So I see we have a few questions here. First one, let's start with the folks at Superluminal. So Mark, Ajay, first question is, how does the data from these alternate sources get into CDD exactly?
Yeah, so the nice thing about CDD, I can answer this one, is just that utilizing common templates across, service providers or biologists, running assays, you can basically either upload to a common spot online the documents of interest, or as we saw James mention in one of his slides, that technically you can even send it directly from the instrument right to a common folder, if you will, which is something that we're actually trying to put into operation currently.
Fantastic, thanks. And if you wouldn't mind keeping you on, we have another follow-up. Someone was not sure. Are you using CDD or LiveDesign to search and track your data?
So basically, we have the option to use both, so that, as mentioned, the kind of data itself is initially stored and is kind of kept in CDD, but utilizing LiveDesign, we can both access the database for the compound information, both structure as well as, compound ID numbers, for instance, and search that, as well as access all the data and the curves and things for either use that can be basically piped directly to LiveDesign when you open said page.
Awesome. Appreciate it, Mark. Thank you. Now, James, if you wouldn't mind, I have a question for CDD here. Can you speak to what else CDD Vault can register? We've seen a few examples today, but can you elaborate on, what else, what other kind of entities or whatnot can CDD register?
Yeah, so absolutely. You can register a lot within CDD Vault. So small molecule, oligos, peptide sequences, proteins, cell lines. The newest thing that we just added, we were actually in DC at the fall ACS. We presented on antibody drug conjugates as our brand new entity that we can do, chemically aware as well. So your biologists will be happy because they can see it in their view, but the atom bond structures are also represented within it. So, and we're always adding new things. So please, if you have something that you're wondering about, by all means, reach out to us and we'll give you the down low.
And Kevin, we have a question here for Schrödinger, if you don't mind. What type of models can you run in LiveDesign? What about GPU-based models like FEP?
It's a great question. Over time, as we've built out this platform, over the last decade or so, the ability to run lots of different types of models, especially very compute-intensive models, has been enabled. So at this point today, you can indeed run FEP Plus through LiveDesign, as well as other very compute-intensive types of models like Jaguar or DFT-based models. And that is a really exciting development because it puts, especially in the hands of like non-expert users, medicinal chemists who might not be trained as computational chemists, it allows them to run these very accurate, but very computationally expensive and challenging models to build. Lets them run it with essentially a button click. There's also the ability for the computational chemist to kind of, let's say, set guardrails around those models to make sure that some of the design ideas aren't really getting out of that chemical space that's appropriate for that series. All of that is available to be built into the specific live design instance that's deployed at every customer site. It really is becoming, I would say, the fundamental sort of design platform for project teams, both computational chemists and medicinal chemists.
Thank you, Kevin. Appreciate it. And unfortunately, that's all the time we have for questions today. I know there's quite a few questions we didn't get a chance to get to, but we will respond to those via a post-webinar follow-up email. I'd like to thank you, our audience, for joining us today for today's webinar, Drug Discovery at Superluminal Speeds. And I'd like to thank our esteemed panelists. Thank you so much for your fantastic presentations and answers as well. All right, everyone have a good day now. Cheers.